Okay, welcome everybody. It's welcome to Notes from the Lab, an ongoing series of podcasts and conversations on the past, present, and future of BCG immunotherapy. My name's Denise Faustman. I'm here in Boston and run the immunobiology labs here at Mass General Hospital. And today's podcast is special because it's going to be about BCG immunotherapy and multiple sclerosis. And obviously, multiple sclerosis is another autoimmune disease benefiting from BCG. And our guest today is Dr. Giovanni Ristori, who joins us from the Department of Neurological Sciences at the University of Rome. And he is really the leader for the last 10, 15 years of trying to get clinical trials moving with BCG in multiple sclerosis. So welcome to our podcast. And I'd love you to just talk about how you got interested in BCG, how you got interested in a generic vaccine, because each of our journeys to why we love BCG is just a little different. So please provide us that background of your journey.
Thank you, Denise, for this kind invitation. It's a pleasure to speak about BCG multiple sclerosis. The first hint that we received was an old paper that was published in Nature in 1958 that demonstrated that the use of a complete Freund adjuvant, that is an equivalent, is a sort of adjuvant therapy, so an equivalent of BCG vaccine, protect from the most used model of autoimmune disease, that is the experimental autoimmune disease. This evidence that may somehow appeared quite paradoxical, because the adjuvant of Freund is used to induce the disease in the animal model, prompt us to hypothesize a use of the vaccine in humans. Moreover, during the 90s in our laboratory, we were studied the T-cell response to autoimmune antigens in multiple sclerosis, convinced us that an effect of benign exposure to microbes, in those time the hygiene hypothesis was put forward, may benefit the course of the disease. So we try a first experiment in vivo experiment, sort of in vivo experiment. It was a little population of patients, and we took advantages of a very informative design for the trial, the so-called single crossover using MRI that was available during 90s, and try to use BCG vaccine in multiple sclerosis patients, receiving the first convincing good signal that BCG may benefit the course of the disease.
Mm-hmm. And one of the unique things about this field, obviously, is what you touched on. There's been a lot of pushback of using a generic vaccine, and most people viewed Freud's adjuvant or BCG correctly as an adjuvant. And so you were going to release all these cytokines and you were going to make people worse. So do you want to talk about that in the context of what we know today?
Yes, it wasn't easy. Many immunologists were afraid of the possibility of worsen the disease course because of the immunostimulant effect of the vaccine, and the fact that BCG may somehow change the cytokine profile toward the TLPR1 profile that may be pathogenic in the immunopathogenesis of multiple sclerosis. But we had, again, evidences that this, to say so, Manichaean distinction between pathogenic and regulatory and protective cytokine profile may be not so easy, not so evident in patients, and then we discover after 90s that as a TNF inducer, the TNF inducer action of a BCG vaccine may be instead be benefit in multiple sclerosis, because in the brain of patients there is a good expression of the receptor 2 for TNF that mediate the reparative phenomenon that underline the beneficial effect of BCG, especially the chronic effect of BCG. I mean the action of the BCG vaccine over time, as we study in the second study that we performed.
Yeah. That's still a point of confusion these days, okay? Because we've gotten more sophisticated at naming these time periods after the vaccine. And there is really absolutely acute innate training, as our colleagues have so elegantly shown. And that's often characterized by changing cytokines, okay? And of course, your and my favorite cytokines, TNF for autoimmunity, ironically, okay? But I think both of us know from our clinical trial data on autoimmunity, those acute effects help, but really you see the big clinical effects 2 to 3 years later, or even greater. They continue to increase in magnitude. And that's not just innate immune training, that's adaptive immune training with big components of T cells and monocytes and B cells. So I think it's hard when people think about the acute vaccine effects to forget that the time course for the clinic is years to get these really significant reprogramming events in effect for better clinical benefit. So that's not just from 20 years ago, that's current, I think. And everybody wants to do the pivotal short trial, right? I want the pivotal short trial. And this drug is not going to give you the pivotal short trial. It may give you the home run, but landing on first base is not going to be sufficient for the regulatory agencies. So very important. I thought of 2 other studies, and you probably know these. There was studies in MS, and I don't know what years these were, that people who got tuberculosis before the age of 12 never got multiple sclerosis. So you start to see those epidemiology studies, and you go, wow, the hygiene hypothesis might really be in play. Probably not as early as 1958, but that data also helped to contribute to maybe there's a signal.
Yes.
Okay,
sure.
So do you want to talk about your published clinical trials of what you've seen?
We were somehow maybe lucky because we observed that our patients in the second trial that we performed, because there was the first one, as I said, was a short trial with a single crossover design. But we were lucky because the same cohort, we had the possibility of studying the same cohort with another a sort of follow-up for 3 years. And in those years, the neuroradiologist put forward the concept of black holes in MRI, magnetic resonance imaging, that is the golden exam used to follow patients with multiple sclerosis. And black hole means loss of tissue, irreversible damage. So we have the possibility to show that patients that had been vaccinated 3 years before had less black holes than patients that did not receive vaccine. And when we designed the second trial, we used patients with an early disease, patients who had the first demyelinating episode, that is the clinically isolated syndrome, the formal definition. And we were lucky because we had the possibility to follow up for 5 years this patient. Yeah. And observing this group, we realized that some patients had a reduction of black hole in their brain. Mm-hmm. And that the conversion to a definite MS, the conversion to definite MS means that you had the second clinical event, was during the 5 years significantly lower in patients vaccinated compared to the group of control. And this gave us the idea that this vaccine may have a note that we use a single vaccination. Mm-hmm. We do not revaccine the patients, at least in multiple sclerosis. Mm-hmm. And this single vaccination may have a long-term action.
Yeah. You know, that, to me, those studies are really important because in the autoimmune field we work in, everybody was trying to use BCG, give one vaccine and look a year later. And, you know, somebody in Israel thought they saw something, and then 3 subsequent studies in France, Canada, the US go, you know, this is ridiculous. We don't see anything. So your study in MS, where you went 5 years, kind of set the gold standard to wake everybody up of this is not a fast drug. You know, and we should never call it a vaccine because the expectation is 4 weeks later, your MS is gone, or 4 weeks later, your diabetes is gone. And it is a long-term thing. And I think that messaging gets lost. And in fact, in the COVID era that we just lived through, I think we saw it relived again of people running out all around the world saying, because BCG is a platform protection from COVID as well. And so everybody was running out to revaccinate, forgetting people had gotten vaccines at birth. People running out saying, well, in 3 months, I'm going to see a difference between BCG and non-BCG. And I kept saying, didn't we learn any lessons the last 10 years about the time course? And, you know, some of our colleagues have had me publish the time course for infectious disease, and then the time course for MS, and the time course for type 1 diabetes, and in adults, unvaccinated. You know, you're talking 3 to 5 years to get those big signals. So I think, again, that's an important point, and COVID allowed us to once again have to raise our hand and say, hey, you forgot, this is not a vaccine for TB that's antigen specific, that's going to confer immunity. It's really significant rewiring of the CNS and signaling pathways. So time course. Now, the good news is it lasts a long time. Okay, so that's the really good news. And so all of us are driven to do these long-term follow-up studies to just see how durable it is, especially in serious diseases like multiple sclerosis. So, okay, so we're getting to the end of the podcast. So where do you think in MS we're headed with BCG clinical trials? What do you see as the pathway for getting this drug to market, at least in multiple sclerosis? And the niche where it'll have a role, yeah.
You know, now it was published from the main MS association in the world, and the Italian one, I must say proudly, is one of these, that is the pathway to MS cure. Mm-hmm. It has 3 components. End of MS, mm-hmm. Reducing MS, reparative phase for patients, as you may know that some group of patients, in spite of very potent therapy, still experience the progressive phase of the disease, mm-hmm. that unfortunately is really disabilitating for people. And so there are these 3 components, and we are expected to try. We are expecting this interim analysis in patients with radiologically isolated syndrome, that is a sort of endophenotype, is a biological MS. If we, as we hope, succeed to have signal in this phase, we will apply for the end of MS to present the BCG vaccine in these people as a form of secondary prevention of this. So because we can expect really to have a great result, because these are people that are people, not patients, because they do not have clinical event. So the BCG vaccine may be ideal in this context, rather than propose to these patients a therapy with pills to be taken day by day, or infusion, intravenous therapy, possible side effects, possible over treatment. It's a great possibility. It's a great space to use this vaccine. In multiple sclerosis, I see at least 3 conditions. Pediatric, because there are pediatric multiple sclerosis. And then there is the so-called late onset multiple sclerosis, that are people that develop the disease after 60.
Really? Okay, that's interesting.
In that case, you may maybe prevent development of in those patients, you can use the disease-modifying therapy that are currently on the market, because they are loaded with very important side effects for people that had 60 years, So if you can propose BCG vaccine, you may also maybe imagine or envision a prevention also of dementia, Alzheimer's disease, the neuroinflammation that characterize the aging, maybe we have no evidences in this context, but I think that in those people, BCG vaccine may have a rejuvenation, is a sort of a rejuvenation approach.
Yeah. And also, we haven't really mentioned it here, but, BCG is very safe. So whether you're talking about immunotherapy and diabetes or immunotherapy and MS, a lot of the therapies are immunotherapy and Alzheimer's disease. A lot of those interventions are quite dangerous. So the other remarkable advantage of BCG, okay, we'll put aside that it's slow to kick in, but we'll also give it major brownie points because it's so safe in large populations. So it can go in the elderly. It also could be used in combination with other things that are less safe. But its brownie points certainly are its safety.
This is the reason why I saw the use of BCG vaccine in people with radiological isolated syndrome, that is a sort of biological MS without clinical manifestation. You can give these people the possibility of living their lives
better
without the load of a therapy.
How old is your population you're enrolling? What's the average age for a clinical isolated syndrome? Yeah.
In clinical isolated syndrome is the classical average age of the population with MS.
Okay.
Radiological isolated syndrome are younger.
A little bit younger. Okay.
Second, third, maximum third decades.
Okay. Got it. Okay. Well, we're so happy to have you here today to talk about multiple sclerosis, and certainly it's a welcoming community as we all try to get more indications for this very safe vaccine in autoimmunity and other indications. So always wonderful to touch base and get the updates myself. Thank you so much for joining me.
Thank you, Denise.
Thank you for joining us today and your many contributions to BCG research and clinical trials as it relates to multiple sclerosis. For all of our listeners, for more information on this podcast series and our BCG clinical trial, please go to www.faustmanlab.org, and all podcasts will be available there for your listening pleasure. Thank you so much today.
Thank you.